the central laser facility in Harwell as part of a small team. And there we go. So at the central laser facility, worked with Dr. Andy Ward, who is an experienced user of a technique called optical trapping, which is also known as optical tweezers. This technique allows you to trap small solid and liquid particles. It works by getting your laser light source to get a laser beam, and then you split the beam into two, and you can control the direction of the beam with a series of mirrors, and then you can control the focus, the intensity of the beam using focusing lenses. Eventually, you'll shoot the beam into objective lenses, which then focus it down to a focal point. And then with this final focal point here, that allows you to trap the solid and liquid particles. And hopefully we've got a video for you. Now this is 2-micron droplet that's getting trapped, and this is a mist of particles. The laser is being filtered out, but that is essentially what we were watching for a few weeks of different things being trapped. Now, we wanted to use this as a technique to replicate the conditions used in a flow tube in the aerosol-assisted solgel catalyst synthesis, which is used to produce high purity nanostructured catalysts without the need for separation. So in AASG, we have a sol which is atomized into droplets containing the precursor solution, which is comprised of the metal salt, the metal alkoxide, and the block copolymer or a different templating agent. So when you heat the droplet, which has been aerosolized, it undergoes, the block copolymer undergoes micellization, forms micelles within the droplet. And then as you heat it more, the gelation reactions occur between the metal salt and the metal alkoxide. This forms an interconnected gel network around the templating agent. And then this is then calcined, and you get a solid porous catalyst material, and the pores are created from the holes in the templating agent. But this has a problem, a challenge which we can overcome, in that it's really hard to replicate the conditions within the aerosol flow tube. Well, it's really hard to study each droplet because it's a really low concentration and they're continuously traveling. But we were able to use the optical tweezers to replicate the conditions of the aerosol flow tube and study operando conditions. So the experimental setup, we had a little nebulizer, and we nebulized our precursor solution. The droplet was then trapped with a precursor solution in it, and we were able to shoot our Raman laser at it. So our Raman laser then gave us a signal based on the species that are present in the droplet, and then we were able to study the changes over time. So we just studied micelles and the gelation. We were also able to add external heat to the droplet to really truly replicate the aerosol flow tube conditions. We studied solutions to synthesize a nickel alumina catalyst, which was synthesized in the literature. So in this instance, our block copolymer was pluronic 3, and our metal salt and metal alkoxide. And then they were mostly dissolved in ethanol, but we also used some nitric acid, which had a little bit of water, and it was a hexahydrate salt, so there was a little bit more water there. We wanted to study each component to see how it affected the reactivity. So we studied the ethanol pluronic as a single solution, added the nitric acid, added the aluminium isopropoxide, and then the whole precursor solution. So let's have a little look at some bulk so we know what peaks we're looking for in the Raman spectra. So the calcined catalyst had nearly no spectra, but we're not really looking at that in the final. But the gel in the yellow here, and the solution, the sol, in the dotted blue here, we have an aluminium isopropoxide associated peak, which was much higher in the solution. And then we have an aluminium nitrate peak, which was much higher in the gel that we formed from the evaporation-induced self-assembly. And we have some CO stretches, which are from the ethanol and the pluronic, which are asymmetric and symmetric. And then we have a lot of different CH associated frequencies, all to do with the ethanol and the pluronic. Here, we're looking at our first, most simple droplets. So on the left, we have ethanol pluronic, very simple, and then acidified ethanol pluronic nitric. This is the first 10 scans, which have been averaged, and the last 10 scans in the blue. So as you can see, the spectra is just dominated by the CH vibrations. There's not much else we see, there's not much else we expect to see. But from this, we can see the ethanol pluronic was held for 4 hours, so it was a really long time, but the addition of acid made it stable in the trap for a shorter period of time. This is attributed to acid destabilized micelle formation, which has been written about in the bulk solution, but this is a novel observation in the droplets. So it's significant that this also occurs in the aerosol-assisted solgel synthesis process, as well as the bulk solgel synthesis. Next, we'll have a look at the addition of the aluminium isopropoxide and then the full precursor solution. So this is without the nickel nitrate, and this is with the nickel nitrate. So we can see there's the appearance of this really big new peak. You can see this. This before we talked about was the aluminium nitrate associated peak. And similarly, the one without the nickel nitrate was much more stable, and the trap was held for a long period of time. And the one with the full precursor solution was less stable in the trap and could only be held for 50 minutes or so. Now, here what we're looking at is each of those peak intensities tracked against time. Now, in this instance, we're sort of substituting the space domain, which would be in the aerosol flow tube, with the time domain to give us a really operando investigation of the single droplets. And looking here at the one without the nickel nitrate, you can see the aluminium nitrate stretch increases substantially, levels out after around 2 hours, but it does increase overall. And you can see these CH stretch vibrations decrease. This is just associated with the evaporation, which we were seeing before in the simple droplets. But it's really significant that we are seeing micelle formation, but no evidence of the true gelation, because there's still lots of aluminium nitrate peaks. There's aluminium isn't bonding with itself in a true way. For the full precursor solution, we just see all peaks that are decreasing rapidly. So this is an indication that there is some actual reactions occurring between the aluminium nitrate species and the nickel nitrate species that we've added in, as well as the evaporation. It's also less stable, probably because of these reactions that are occurring. So the nickel nitrate we have determined here is required for the gelation reactions under isothermal conditions. But, now here we're looking at exponential decay analysis fitted according to, that's my cursor, this equation here to find out the rate of decay for each of the intensity of the peaks, because this allows for the better comparison of the decay rates from one spectra to another. Here we can see this is the CH stretching peak, which decreases over twice as fast in the full precursor solution compared to the one without the nickel nitrate. And then it's similarly over twice as fast for the CH bending. So they're both representative of that evaporation. And you can see that measurably the evaporation is faster regardless of these gelation reactions that we've seen occurring in the full precursor solution. Now we're looking at the really true representation of the aerosol-assisted solgel synthesis flow tube, because we've added heat to the equation. So this is really a novel investigation into the early heating of trapped droplets. And What's most interesting is we see the opposite behavior. If you remember for the one without nickel nitrate before, the only peak to decrease were the CH associated peaks, whereas here the CH associated peaks aren't decreasing, and the aluminium nitrate peak is the only peak to decrease. And in the one with the nickel nitrate, we're just seeing the enhanced behavior. This is what we expected because there's more heat, there's more activation energy, things evaporate faster, and things can react faster. So let's look a little bit more closely at that without nickel nitrate droplet, studying each peak intensity against time. So the only peak to decrease, as we saw, was the aluminium nitrate peak. And here you can really see that the temperature increase, whereas you saw the early temperature, there's a little bit of evaporation. As soon as we start ramping the temperature here, the evaporation is completely hindered. So this is what we would speculate to be an encapsulation type mechanism, where the gel, the pluronic, not the micelles, or the gel is encapsulating around the entirety of the ethanol and preventing it from evaporating, which is a really, really interesting finding. And then, of course, we've got more activation energy, which could allow for these gelation reactions to occur in the aluminium groups. And then, like we said before, we couldn't heat this droplet up as fast because it just reacted so fast. It really wanted to do those gelation reactions as soon as you've got that nickel nitrate in there. So normally in the literature for aerosol-assisted solgel synthesis, they assume the reaction mechanism based on the bulk observations. And the assumption would be that they would get the hydrolysis first for the aluminium isopropoxide, where there would be an acidified isopropoxide group that would undergo a conjugated addition elimination reaction with water. And then you'd get left with your aluminium hydroxide group. This aluminium hydroxide group could then undergo polycondensation reactions with other aluminium hydroxide by performing reactive attack of the other aluminium, stabilized transition state, and kicking out some water. And then you'd get a full gel network of lots of aluminiums bonded together through this oxygen. But we can't see any evidence of this in our trapped droplets because we can't see any aluminium hydroxide. We can't see any of the aluminium isopropoxide groups. We don't really have enough water in our droplets. And this is the same synthesis medium that the other people were using to actually initiate the hydrolysis mechanism. Therefore, we've proposed an alternative reaction mechanism based on our investigations. And this proceeds by the initial attack of the isopropoxide group onto one of the nitrate groups, the acids, which gives a nice leaving group. So then you can get a reactive, positively charged aluminium, which can form an aluminium nitrate. And we saw that aluminium nitrate forms in the droplet without the nickel nitrate, just increased over time to a limit. Because then you'd get your aluminium nitrate, and this would be maybe with more nitrates on, but this will be what we're observing in the droplet without the nickel nitrate. Then, as soon as we add the nickel nitrate, there's some reactivity happening in isothermal conditions. Let's say the nickel is attacking the aluminium, and then the nitrate is acting as a leaving group. This will allow for a poly gelation reaction where more nickel combined to more aluminium combined to more nickel, and you get your extended gel network forming. So in summary, we have successfully optically trapped droplets containing each component in the solgel synthesis medium, which replicates the Operando conditions for AASG, and these have been studied in Raman spectroscopy. And this technique has allowed us to validate or oppose what has been assumed about the reactivity in AASG based on bulk solgel synthesis. And we have evidence for micelle formation, as well as the micelle formation being influenced by things in the reaction mixture and the effective acids. And we have also found evidence for gelation reactions and proposed an alternative reaction pathway, given that we didn't see what was expected for bulk solgel synthesis. So thank you all for listening. I'd like to acknowledge everyone involved in the research, particularly Dr. Andy Ward for his expertise and the Central Laser Facility for funding the research time. Hopefully we have a few minutes for some questions.
Yes, we have time for questions. If you'd like to type your questions in the chat and I will read them out. I have possibly what's a little naive question, and I hope it's not an evil question. So you've obviously shown this optical trapping combined with Raman. Do you think there's scope to combine it with other techniques, sort of FLIM or Kerr-gate Raman for samples that might fluoresce? Or
yeah, oh, of course there is. Yeah, it's used a lot in biological samples, and they even use things like Fourier transform infrared and, like you say, corrugated Raman. But yeah, absolutely, you can. Okay. It gets a little bit harder depending on what sort of intensity laser you want to shoot, because it can destabilize the droplet. But you can balance it. That's why you have the beam splitter, because then you can control the intensity of one beam to be able to shoot a different laser source at it.
Okay, thank you. Asked, how did you produce the heat?
We had a little. It was just like your oven heater. It heats with electricity, and it was on the cell. So we have this little cell here, and the droplets coming in, and it's getting trapped. And either side of that, we had a little hot plate type thing. It's really tiny, and we just had it of a known voltage that we applied to it. But we had to like standardize the amount of heat you were getting from what voltage you put in.
Okay, I hope that answers your question, Donnie. Are there any more questions? I have another one, which is, obviously you've shown this for sort of a solgel type synthesis. Is there any other sort of syntheses you think this might be applicable to, this optical trapping? Because obviously it's quite specialised.
Yeah, I think that it's for any process which uses a flow tube, because it can really replicate those conditions, especially if you've got a more stable, less volatile reaction mixture, more solid or something. So it really useful in studying the synthesis of catalysts that use flow tubes, or even reactions that occur in flow tubes, just to replicate the conditions.
Okay, that sounds really interesting. Vidal has a question, sorry if I mispronounced your name. He's regarding the morphology of the catalyst you're producing. He didn't see any morphological images. Do you have any idea what morphology they're coming out in?
No, it would just be, we didn't actually make any with the aerosol flow tube, because we don't have an aerosol flow tube. We made them in bulk, and we studied the droplets replicating the early conditions in the aerosol flow tube, but we didn't make any by that way. But if you just make them,
it's such a small sample that you're making in the optical trapping that you can't really do anything with them.
Exactly, yeah.
Need to get a micro TM. That's the next. And Donnie asks, how did you detect the temperature inside the droplet?
Yeah, so we couldn't do that. We could not do that. We could only standardize the amount of voltage and the amount of temperature we expect the position that the droplet would be in to reach.
Okay, thank you for that. If there's no more questions, I'm sure there is a virtual round of applause going around. Thank you very much for an excellent talk.